Here's the gas exchange system. We have the air line that we must turn on. Air will then flow through into the flow meter, which adjusts the rate of airflow throughout the system. Air will then flow into the leaf chamber, which is located here. There's a light source on top of it, and then the leaf fits inside the little chamber here. To take a reference, we want to make sure that the chamber is empty, and then we want to seal it with these screws. Air will then flow out of the chamber into the relative humidity and temperature sensor, which allows the computer to record humidity and temperature of the air that's going through the system. And then the air will then go through the CO2 analyzer for the CO2 concentration to be measured. To open Logger Pro, it's found under Course Applications. Scroll down until you find Logger Pro 3.15. Open that. From here, we want to open up the experimental file for the sensors. So under File, Open, go to this PC, Windows, File 312, experimental file for new sensors. Open. So here we need to confirm what sensors are plugged in. For this part, because everything is in order, we can just click Connect. We want to verify that we have our CO2 analyzer as the first one, the raw voltage, which is the light, the relative humidity sensor, and the temperature sensor. To calibrate the light source, we first open up the mini window by clicking here. We will then select channel 2, raw voltage, which is the light source, and select Calibrate. At the Equation tab, we find the intercept and slope. The values here are found on a sticker on the light source itself. So we will input the values. Each instrument has its own unique identification of intercept and slope, so make sure you use the appropriate one. Now we will click Apply to make sure that the changes take effect. Next, we click Calibration Storage. We want to ensure that the experiment file is selected, and then click Done. We can then close out of this mini window. Now we want to verify that our sensors are properly recording and showing good information. So here we have our CO2 concentration. It should be around 400 to 410. Temperature should be room temperature, so about 23, 24 degrees. Relative humidity, and the light. The light information here is based on the position of the dial on the light source. So anything negative, you can just assume is zero light intensity. To start collecting data, we will then click Collect. Here we will see data start to compile. We can also adjust the y-axis range on these figures by clicking the top value and inputting our own manual version value, as well as the minimum by clicking on here. Now we have a better view of our data as it fills out. To put a leaf inside the chamber, we first want to apply vacuum grease along the gaskets to ensure a proper seal. So we want to open up the chamber, get our vacuum grease, a small amount on your fingertips, and just apply it along the gaskets. You want to make sure that you don't get it inside the gasket because that's where the air flows along. And we don't want to clog up any of the flow. So to put our leaf inside the chamber, we want to make sure that the plant is properly adjusted height-wise, either through the gas exchange, the chamber, or the plant itself. That way we're not producing any stress on the plant. So we put our leaf inside the chamber. We want to fill up the entire chamber if possible, close it, and then seal it using all 3 of these screws. Once all 3 screws are in place and tightened so that there are no air leaks, we want to make sure that there are no air leaks, so we do a blow test. And that is by blowing air around the chamber, just using our mouths. On the computer screen, we would watch the graph and see if there is a spike in the CO2. If there is a spike, that means we have an air leak, and we must make sure that the chamber is properly sealed, either by applying more grease or double checking the screws and the tightening. For the first light intensity, we want to turn the dial. Here, as we turn it, we want to pay attention to this LED information box. We want to set it to about 200. And this will be the second data point for the light response curve. For the next one, we will then bring up the light to 400. And we will continue increasing the light intensity as in the protocol. For each light intensity, we want to have a stabilization time of about 4 minutes. Data will then be recorded manually by the students by writing down the value here at the end of the 4-minute stabilization period.